my name is Piper. Today we're going to be exploring an important part of all living things, the cell. All organisms, or all living things, are made up of cells. The cell is the basic functional instructional unit of every organism. It is the simplest collection of matter to be alive. Our bodies are each made up of 32.7 trillion cells. How is it possible that there are so many cells in your body? Cells are very, very, very small. In fact, cells are so small that we can't see them with our human eye, and scientists have to use microscopes to be able to see them. Just as your body is composed of many organs that allow you to function properly, like your stomach, your heart, your lungs, each cell is composed of many organelles that allow them to function properly. Each organelle has its own job, and by working together, they form a functioning cell. It's just like when you help your family do the chores or make dinner. Everyone does something to pitch in. Today, we're going to be exploring an animal cell and make a model of it. Every item in our model today will represent an organelle. You can use the various fruits and candy as I used, or you can be creative and come up with your own. At the end of our experiment, you can even eat your model. When making our cell model, the first thing we need to build is the plasma membrane and the cytoplasm. In our model, the plastic bag or bowl will represent the plasma membrane, which protects the cell. Just as a plasma membrane encloses a cell, the plastic bag or bowl encloses the jello. The cytoplasm of a cell is the jelly-like material that makes up the inside of the cell where the organelles are located. What in our model do you think represents the cytoplasm? Let's think. If you guessed the jello, then you are correct. Let's prep the first few steps together.
Purchase a Jello set at the nearby store. We recommend following the instructions from the actual Jello set. The way we did it was we boiled water, added the gelatin, and then mixed the gelatin in cold water. Then we recommend refrigerating it. This is really important. Only refrigerate it until it is half solidified. You should still have a pretty liquidy substance. This is when you're gonna add your organelles. You can either pause the video here and wait until it is half solidified, or you can watch the whole video through and then add your organelles after you've watched and it is half solidified.
While we wait, let's talk about organelles and what candies we want to use to represent each one. I recommend filling out our provided coloring sheet or making your own cell drawing to keep track of each organelle. As I talk about each organelle, color it in on our provided coloring sheet or add it to the diagram you're making. We can do the first one together. Let's look at number 1 on the coloring sheet, the plasma membrane. The plasma membrane protects the cell. The plasma membrane is kind of like the walls of your house. Just as the walls of your house protect all of the belongings on the inside, the plasma membrane protects the jello, or cytoplasm, and all of its organelles on the inside. In our model, the plastic bag or bowl represents the plasma membrane. Color in the plasma membrane and write that the bag represents it on the line below. Now, let's quickly jump to number 9 on our coloring sheet, the ribosomes. Ribosomes are one of the smallest organelles in our cells, but they do big work. Who is the chef in your household, or who does the cooking? Ribosomes are like the chef in your house. Just as a chef cooks or makes meals, ribosomes make proteins for the cells. Moving on, let's discuss number 2 and number 3 on our coloring sheet, the smooth endoplasmic reticulum and the rough endoplasmic reticulum. Endoplasmic reticulum is a pretty tough word, so let's try saying it together. Endoplasmic reticulum. The endoplasmic reticulum has 2 portions, the smooth portion and the rough portion. Let's refer to the smooth endoplasmic reticulum as the SER and the rough endoplasmic reticulum as the RER. On our coloring sheet, the RER has a bunch of dots on them. Does anybody think they know what those small dots are? If you guessed ribosomes, you are correct. The RER has ribosomes attached to its outer surface, making it look rough under a microscope. The rough endoplasmic reticulum, or RER, works to produce, store, and export, meaning send off, proteins. It makes sense that ribosomes are attached to the RER because ribosomes work to produce proteins. The SER does not have any ribosomes on its surface, making it look smooth under a microscope. The SER works to produce lipids or fats. Now on to number 4, the mitochondria. The mitochondria acts as the powerhouse of a cell. They work to convert energy from food and nutrients into energy that the cell can use. The mitochondria is kind of like a power plant. A power plant generates energy that an entire city can use, just as mitochondria generate energy that the entire cell can use. Let's move to number 5, lysosomes. Lysosomes are organelles that function in digestion and removing waste from cells. Lysosomes digest excess or extra food and bacteria from the cell. Lysosomes are kind of like recyclers. Just as recyclers remove your trash from your house, lysosomes will remove trash from the cell. Now, on to numbers 6 and 7, the nucleus and nucleolus. The nucleus contains our DNA and our genes, and therefore provides cells with the instructions and commands it needs to function. The nucleus of a cell is a lot like a boss. It directs all of the cell activities. The nucleolus is a dense area inside of the nucleus. The nucleolus makes ribosomes and subunits from mRNA. The nucleolus is a lot like the secretary of the boss. The secretary has all of the papers, maintains the boss's files, and sends the boss's emails. Let's jump on to number 8, centrioles. Centrioles are organelles that help with cell division, a process in which a cell divides and replicates itself into 2 daughter cells. Centrioles are kind of like a copy machine. Just as a copy machine makes a bunch of copies of papers, centrioles allow cells to make a bunch of copies of themselves. To number 10, the Golgi Apparatus. The Golgi Apparatus looks a lot like a stack of pancakes, but it is very important, for it involves sorting and packaging proteins. It also transports lipids or fats outside of the cell and creates lysosomes, the recyclers that we discussed earlier. Because the Golgi apparatus involves transporting molecules throughout and outside of the cell, we can easily compare the Golgi apparatus to a post office. A post office receives mail, sorts mail, and then sends mail to its desired location. Similarly, the Golgi apparatus receives molecules, changes them, sorts them, and then sends them to different areas within the cell. Now, on to the last organelle of our coloring sheet, number 11, the cytoplasm. We discussed the cytoplasm earlier. Do you remember what we used in our Jello cell model to represent the cytoplasm? If you guessed Jello, then you are correct. The cytoplasm is a gel-like liquid inside the cell. It works to keep organelles in their place and fill the entire cell. Now that we have covered all of their organelles and their functions, and we have filled out the coloring sheet, let's decide on the various fruits and candies we want to use in our Jello cell model. Try to be creative and use candies or fruits that you can find in your house. Now, what candies or fruits could we use to represent the organelles? What candies or fruit look like certain organelles? What candies or fruits could represent the function of an organelle? Here's an example. We could use normal gummy worms to represent the SER because they look smooth, and sour gummy worms with the sugar on top to represent the RER because they look rough. Once you have come up with the candies, make sure to write them down on your coloring sheet. Now, let's put all of the candies into our gel model. Now that we have all of our organelles into our cell model, let's put our model back into the refrigerator to let the Jello to completely set. After an hour or two, you can remove your cell from the refrigerator and place it into a plastic bag to examine your 3D model. Once it's ready, try to see if you can identify the different organelles in the cell. Act as a biologist and teach your family about the organelles that you learned about today and their function. Also, take a picture of your coloring sheet and send it to Curieus@Curieus.org to win some potential prizes, such as Curieus merchandise.
Now that we have finished our experiment, we're going to discuss an important scientist, Jennifer Doudna, that created a technology that can be used to edit eukaryotic cells, which are just cells that have a nucleus and organelles, just like the one we just made. This technology is known as CRISPR-Cas9. CRISPR-Cas9 edits the genome of cells by changing DNA sequences and modifying gene function. I know that sounds complicated, but it just means that it changes the way your body functions. CRISPR-Cas9 can correct genetic defects, treat and prevent the spread of disease, and also can improve crops.
Thank you guys, and we'll see you tomorrow.